Okay. So the paper I plan to do today is Winter 2025, paper 2, variant 2. So the wall of the small intestine is highly folded to form villi. Between the villi and enfoldings, between the villi are enfoldings known as crypts of lubricant. So over here we see enfoldings. They are basically known as the crypt of lubricant. Figure 1.1 is a diagram of a section through a single villus and a crypt of lubricant. Figure 1.1 shows that the sorry. Figure 1.1 shows that the epithelium of the villus contains mainly globule cells and cells known as enterocytes. So the epithelium basically consists of enterocytes and it consists of goblet cells. Both cell types have microvilli on the apical surface. Apical surface is the surface facing the gut lumen. So ये हमारे पास gut lumen है. So इस वाली surface को, this is the apical surface. And on the apical surface of all the enterocytes and goblet cells, there are microvilli present. Goblet cells are involved in the production of mucus. Enterocytes are adapted for the absorption of the soluble products of digestion. These products enter the circulatory system. A student incorrectly stated that an enterocyte has many cilia on its apical surface. Explain the difference between a cilium and a microvillus. So a cilium is basically made up of microtubules which are arranged in a 9+2 arrangement, whereas microvillus, microvilli, they're made up of actin filaments. So that's the difference. Yeah. So cilium is made from microtubules which are organized in a 9+2 arrangement. Microvillus is made up of actin filaments. Okay, so just one role of the mucus produced by the goblet cells of the villus. तो हमारे पास क्या कहते हैं जो हमारी apical surface है, that apical surface basically has mucus lining on it, right? हमारे पास जो mucus होता है, mucus is basically a thick fluid. अब जब हम बात करते हैं mucus की in the epithelium of the small intestine, तो हमारे पास क्या कहती हैं? It basically is there to trap pathogen if any pathogen has entered through the food that we are consuming. So उसको trap करता है या फिर हमारे पास क्या कहते हैं जो mucus है, it's a fluid, right? तो वो क्या करता है? It basically reduces friction or any wear and tear जो कि friction की वजह से होती है. So it basically reduces friction when food particles are moving through the gut. So हमारे पास जो gut में कहीं पर भी mucus होता है, क्योंकि food particles are solid particles and they're obviously brushing against the walls of the gut. तो हमारे पास क्या कहते हैं, it is possible कि अगर mucus नहीं होता तो हमारे पास बहुत ज्यादा friction होती है जब move कर रहा होता हमारा food particles, solid particles through the gut. So the mucus has a role of even reducing friction when solid particles or digestive material is moving through the gut. So we can say traps pathogens. और अगर कोई ऑल्टरनेटिव हम लिखना चाहे तो ऑल्टरनेटिव could be reduces friction for the movement of digested substance. In figure 1.1, blood vessel X delivers blood to the capillary network. If it is delivering the blood to the capillary network, then definitely has to be oxygenated blood. Where tissue fluid is formed, some of the fluid passes back into the capillaries and then into the venule. Blood vessel X receives blood from an artery. So blood vessel X is receiving blood from the artery and it is delivering the blood to the capillary. Name the type of blood vessel represented by X. So X would be an arteriole because arterioles are the connecting bridges between the artery and the capillaries. Anita, they have mentioned that the blood vessel X receives blood from an artery. So it is a blood vessel which is not an artery. Instead, it is receiving blood from an artery and it is delivering the blood to the capillary network. So the blood vessel between the artery and capillary, or the connecting bridge between the artery and the capillary, is the arteriole. Describe the formation of tissue fluid in the capillary network of the villus. So basically, blood enters the arterial end of the capillary at a high hydrostatic pressure, and thus the components, the small components of the blood, such as glucose, amino acids, are filtered out of the blood with the blood plasma through the fenestrations. Fenestrations are basically the gaps or the pores in the endothelial walls of the capillary. In response to the presence of compounds in the gut lumen, enteroendocrine cells synthesize and release peptides, short chains of amino acids that are cell-signaling molecules. One of these cell-signaling molecules is known as GLP-1. GLP-1 initiates a number of responses in different body cells. Some of these responses include the increase in release of the hormone insulin from the cells in the pancreas, the decrease in release of acid from cells in the stomach. Outlined in sequence, the main stages involved in the process of cell signaling by GLP-1. Now, हमारे पास ये कहते हैं cell signaling में basically there are three stages. One stage is basically the production of the cell signaling molecule from the targets, from the site of origination or the site of production. Site of production हमारे पास क्या है? हमारे पास site of production enteroendocrine cells हैं. So the GLP-1 is produced and secreted by the enteroendocrine cells. It travels via the bloodstream to the target cell, which would have a receptor complementary to GLP-1. The GLP-1 would bind to the receptor and trigger a sequence of responses within the target cell. So we have to talk about the movement of the cell signaling molecule via the bloodstream, its movement towards the target cell, and its binding with the complementary receptor to gain our 3 marks. Pannette cells, which are secretory cells, are located between intestinal stem cells at the base of the crypt of lubricant, as shown in figure 1.1. Figure 1. A Paneth cell has a very different appearance to an intestinal stem cell. Figure 1.2 is a transmission electron micrograph of a Paneth cell. So this is basically our Paneth cell. Surrounded with obviously intestinal stem cells. इसके surrounding में intestinal stem cells हैं और इनके बीच में हमारा Paneth cell है. Pinnet cells are formed following the mitotic division of an intestinal stem cell during a cell cycle. Complete the cell cycle shown in figure 1.3 by naming in sequence the stages of mitosis. This sequence is PMAT, which is prophase, metaphase, anaphase, and telophase. One of the functions of a Paneth cell is to synthesize and secrete peptides and proteins that act against pathogens in the gut lumen, state and explain the evidence visible in figure 1.2, which suggests that a Paneth cell is a secretory cell and synthesizes many peptides and proteins. Now, if it is a secretory cell, what evidence do we have in the figure to tell us that a Paneth cell is a secretory cell? A secretory cell is basically a cell which transports substances, which forms substances and then transports those substances outside of the cell. And obviously, हमारे पास substances as bulk transport move out करते हैं, so bulk transport में exocytosis होता है. Over here, we can see कि हमें बहुत सारी vesicles दिखाई दे रही हैं. 1, 2, 3, 4, and so on and so forth. हमें बहुत सारी vesicles of different sizes दिखाई दे रही हैं. Now हमारे पास क्या कहते हैं? The presence of many vesicles confirms that this is a secretory cell, and these vesicles would basically fuse with the cell membrane and release their contents outside of the cell via exocytosis. So secretory cell होने का हमारे पास जो evidence है वो presence of a lot of secretory vesicles है. फिर उन्होंने हमसे second evidence मांगा है कि how can we say, looking at this particular cell, that it is involved in the synthesis of peptides and proteins. If we look closely, can you see कि हमारे पास rough endoplasmic reticulum बहुत सारे हैं. आपको ribosomes दिखाई दे रहा है on the endoplasmic reticulum. So the presence of RER, a lot of RBR or an abundance of RER within the cell suggests that this cell is involved in the secretion of peptides and proteins. फिर हमें क्या कहते हैं, माइटोकॉन्ड्रिया भी नजर आ रहे हैं. दिस इज अ माइटोकॉन्ड्रिया, दिस इज अ माइटोकॉन्ड्रिया. ये दोनों भी हमारे पास माइटोकॉन्ड्रियाज हैं. The presence of mitochondria protein synthesis is essentially an active process. It requires ADP. So many mitochondria within the cell suggest that they are actively playing a role in synthesizing ADP, which is needed in the protein synthesis process. Okay. There are many secretory vesicles within the Paneth cell, suggesting that they contain substances which will be secreted outside of the cell via exocytosis. The Paneth cell has an abundance of rough endoplasmic reticulum, which is responsible for protein synthesis, mitochondria, are also present for ATP synthesis, which is needed during protein synthesis. So one mark for secretory vesicles, one mark for the presence of rough endoplasmic reticulum, and another mark for mitochondria. Explain why Bennett cell has a very different appearance to an intestinal stem cell. Obviously, an intestinal stem cell can differentiate into any cell type. It can differentiate into an enterocyte, into a goblet cell. तो किसी भी intestinal cell में stem cell differentiate कर जाता है, right? Because initially वो undifferentiated होता है. So yeah, you can also go use the word of specialized. So specialized हो गया, differentiated हो गया, आप वो use कर सकते हो. We would say that Paneth cell is differentiated or it is specialized compared to a stem cell, which is undifferentiated or it is unspecialized. So Paneth cell, let's move towards the next question. Cholera is an infectious intestinal disease caused by a bacterial pathogen. Name the species of bacterium that causes cholera. So that is Vibrio cholerae. The World Health Organization, WHO, recommends a number of different approaches for the prevention and control of cholera. Two of these are authorities should provide access to safe drinking water. Individuals and communities should practice preventive personal hygiene. Suggest can explain how these two approaches help in the prevention and control of cholera. Now, cholera is transmitted through the fecal oral route. If we are drinking contaminated water, it is likely that we ingest the cholera bacterium, and therefore drinking safe water would prevent the entry or the ingestion of Vibrio cholerae within our system. To practice preventive personal hygiene basically means that every time we are using the washroom, we end up washing our hands to ensure that any feces is not present on our hands. And if feces are not present on our hands, they would not contaminate food or water, through which the transmission chain of cholera would be propagated. So cholera spreads via fecal oral route and is present in contaminated water. Drinking clean and safe water would ensure that that bacterium causing cholera is not ingested. Practicing preventive personal hygiene include frequent washing of hands, especially after using the toilet to ensure that the feces are not present, food or water. Mass vaccination is mass vaccination using an oral cholera vaccine can be carried out in situation where there is a high risk of people developing the disease. Passive immunization involves transferring antibodies into a person for the prevention or treatment of an infectious disease. Some of the infectious diseases for which passive immunization is available use monoclonal antibody. Passive immunization for cholera using monoclonal antibody could be available in the future. Complete table 2.1 to compare an oral cholera vaccine and passive immunization for cholera. Fill in the empty box in row 1. Circle the correct answers from the choices given in rows 3, 4, and 5. Okay, so in an oral cholera vaccine, the component causing the desired response is an antigen, or we can say an attenuated pathogen. Attenuated pathogen is basically the pathogen in its weakened form from which we remove the disease causing ability. Now, when we look at rows 3, 4, and 5, stimulates production of memory lymphocytes. Now, because there is an antigen which is being injected in the body, there would be the stimulation of B cells, which would differentiate into plasma cells and memory cells. In passive immunization, because there is no antigen being injected in the body, there would be no immune response, and thus there would be no production of memory lymphocytes. Length of time needed to have an effect. Obviously, हम जब vaccine के through when we inject a pathogen in the bloodstream, it takes a time for the antibodies and the memory cells to form, because first we have an innate immune response, which is basically phagocytosis, and then we move towards the adaptive immune response, which includes your humoral immune response, which we call the antibody-mediated immune response. So it takes longer. However, because we're directly injecting antibodies in the passive immunization, it would take a shorter time for the response to be produced. Duration of immunity, a vaccination gives you a long-term immunity, so it gives you longer immunity. Whereas the duration of immunity, when we're just injecting antibodies, it is shorter because eventually the antibodies would break down. The production of monoclonal antibodies for treatment involves the formation of hybridoma cells from two different cell types. Name the two types of cells that fuse to form a hybridoma cell and explain why this fusion is necessary. So हमारे पास क्या कहते हैं हम? We fuse the myeloma cells, which are cancerous B cells, with the myeloma cells are fused with plasma cells. And why is this needed? It is needed because plasma cells cannot divide via mitosis, whereas myeloma cells can divide via mitosis. Plasma cells can produce the desired antibodies. So plasma cells can produce the desired antibodies. However, they cannot divide via mitosis. Myeloma cells can divide via mitosis. Therefore, fusing them together forms a hybridoma cell which can divide and produce desired antibody. Now let's move towards question number 5. A plant is described as a mesophyte. A plant that is described as a mesophyte has evolved to grow in conditions that do not normally experience water stress. तो हमारे पास mesophytes basically ऐसे plants होते हैं जो grow करते हैं उन conditions में जहाँ पर water availability होती है, excess में water present होता है. They do not experience water stress or low availability of water, basically. Figure 5.1 is a diagram of a cross section with a leaf of a herbaceous like cotyledonous mesophyte. We complete figure 5.1 by naming structure A and the tissue layer B. So the structure A is basically a layer above the upper epidermis, so that is the layer of cuticle. इसको हम waxy cuticle भी बुला सकते हैं, cuticle भी बुला सकते हैं. B is basically a layer of spongy mesophyll cells. B हमारे पास हो जाएगा spongy mesophyll cells. Okay. They're basically asking us to name the tissue layer, not the cells. So spongy mesophyll tissue, लिखेंगे, not spongy mesophyll cells. In xerophytes क्या होते हैं? Plants adapted to live in water stress. In xerophytes, some of the structural features shown in figure 5.1 are modified as adaptations for surviving conditions of water stress. Complete table 5.1 to show how the structural feature listed may be modified in the leaf of a xerophyte. Each feature should have a different example of a modification. Structure A. Waxy cuticle. How is a waxy cuticle different in a xerophyte? Would it be the same or would it be different? And if different, then how? Yeah, exactly. So, waxy cuticle is thicker to prevent वाटर लॉस. फिर अपर एपिडर्मिस, हमारे पास अपर एपिडर्मिस जो हमारे पास मेसोफाइट्स होते हैं उनमें सिंगल लेयर होती है अपर एपिडर्मिस की. ऑन दी अदर हैंड हमारे पास जीरोफाइट्स में मल्टी लेयर्ड एपिडर्मिस होता है. हमारे पास 2-3 लेयर्स होती हैं अपर एपिडर्मिस की सेल्स की, so that water loss through the upper surface इतना easily ना हो. Multi-layered upper epidermis. फिर लोअर एपिडर्मिस में हमारे पास फ्यूर स्टोमेटा होते हैं या हमारे पास संकन स्टोमेटा होते हैं, ट्राइकोम्स प्रेजेंट होते हैं. तो इनमें से हम कोई सा भी एक अडेप्टेशन लिखते हैं. फ्यूर स्टोमेटा. Okay, so we have done 3 questions. Question 1, 2, and 5 yesterday. We'll do 3, 4, and 6 today. The gene LCT codes for the enzyme lactase. In babies, lactase synthesis is necessary for digesting lactose, the sugar found in milk. So lactase is needed for lactose digestion. Another gene, MCM6, has introns that have a regulatory role in the expression of gene LCT. Regulatory role in the expression of gene LCT basically means that they basically control the amount of times, regulate the amount of times the gene LCT would be expressed. So they're basically regulating the transcription of the LCT gene. Gene MCM6 codes for a protein that has no involvement in lactase synthesis. As children got older, the MCM6 introns are responsible for a decrease in lactase synthesis. So, with age, the MCM6 introns are responsible for a decrease in lactase synthesis. This decrease in lactase synthesis is also known as lactase non-persistence. LCT and MCM6 are located on the same chromosomes in humans, suggest differences between gene LCT and gene MCM6 other than their locations and different positions on the same chromosome. Obviously, if there are two different genes, the sequence of nucleotides would be different. Because if sequence of nucleotide would have been the same, they would have been the same genes, not different. So, sequence of nucleotides of gene LCT and gene MCM6 would be different. Then it is also possible the length, which means how many nucleotides make up a single gene, would be different. So हमारे पास क्या कहती हैं? Different lengths, the two genes would have different lengths. Now we know that LCT codes for an enzyme. So we can say that the enzyme LCT is coding for an enzyme, whereas MCM6 is coding for a different protein. So LCT gene codes for an enzyme, lactase, whereas the MCM6 codes for a different protein product. With reference to the process of lactase synthesis, explain the relationship between a transcribed strand and a primary transcript, a primary transcript and a messenger RNA. So we have to tell how transcribed strand and a primary transcript are related, and then we have to explain how a primary transcript and mRNA are related. So हमारे पास DNA के दो strands होते हैं. We have two strands of the DNA. In transcription, only one of the two strands is used, which is known as the transcribed strand. From the transcribed strand, we form the primary transcript. So हमारे पास transcribed strands, this is our transcribed strand, this is our primary transcript. So the transcribed strand is transcribed, or it undergoes transcription for a complementary primary transcript to be formed. Now, this primary transcript has both the introns and the exons. The introns from the primary transcript has to be removed via the process of splicing so that an mRNA can be formed. So, हमारे पास primary transcript की splicing के बाद या modification के बाद eventually mRNA बनता है. So transcribed strand is transcribed to form a primary transcript, a complementary primary transcript, whereas the primary transcript undergoes a post-transcriptional modification, which involves splicing of the introns to form mRNA. So we'll write that down. The transcribed strand is the DNA strand which undergoes transcription to form a complementary primary transcript. The primary transcript undergoes post-transcriptional modification in which introns are removed via splicing to form mRNA. Clear? Any questions so far? A mutation in a regulatory intron of MCM6 allows lactase synthesis to continue. This is known as lactase persistence. In this mutation, the number of nucleotides in the intron remains the same, but one of the nucleotides is different to the original nucleotide. State the type of mutation that is the cause of lactase persistence. So हमारे पास तीन तरह की mutations होती हैं. बेस एडिशन, बेस डिलीशन, बेस सब्स्टिट्यूशन. अगर हमारे पास बेस एडिशन या बेस डिलीशन हुई होती तो हमारे पास क्या कहते हैं, नंबर ऑफ न्यूक्लियोटाइड्स wouldn't have been the same. It would have either increased in the case of base addition or the number of nucleotides would have decreased in the case of base deletion. If the number of nucleotides is same, but a nucleotide has replaced an original nucleotide, in that case, base substitution has taken place. Figure 3.1 summarizes the reaction catalyzed by lactase. Draw the ring structure of alpha glucose in the box provided to complete figure 3.1. So AB alpha, which means the OH on the first carbon would be below. Some people with lactase non-persistence may be lactose intolerant. They may have symptoms such as abdominal pain if their diet contains lactose. Lactase supplements, tablets can be taken before milk or milk-based products are consumed to avoid symptoms of lactose intolerance. A student compared the activity of two different concentrations of a lactase supplement using an artificial substrate, ONPG, instead of lactose. A solution of ONPG is colorless, but the hydrolysis of ONPG by lactase releases a colored product. So हमारे पास substrate is colorless, but after hydrolysis, the product is colored. Now, to see how different concentration of a lactase supplement, which helps with lactose digestion, how does that work? उसको understand करने के लिए lactose के बजाय हमने एक different substrate use किया so that we can see how fast the digestion or how fast the hydrolysis is into different concentration of a supplement. Colorimeter को हम use करेंगे इसमें. The student planned to follow the progress of the reaction for each concentration of lactase using a colorimeter. Are you aware of how a colorimeter works, Anna? Or should I explain the basics of colorimeter? I'll show you how a colorimeter looks like. Yeah, it's the one used in the practicals. It also comes up in P1 and P2. So let me show you how it works. Give me a second. मैं picture copy paste कर लेती हूँ. So this is basically how a colorimeter looks like. अच्छा. So हमारे पास होता क्या है? हमारे पास you can see ये basically हमारे पास different टेस्ट ट्यूब होती है. एट अ गिवन टाइम हम यूजुअली एक टेस्ट ट्यूब प्लेस करते हैं. कैन यू सी यहाँ पर एक टेस्ट ट्यूब प्लेस्ड है. मैं थोड़ा सा जूम इन करती हूँ. ओवर हियर कैन यू सी अ टेस्ट ट्यूब इज प्लेस्ड इन द स्पेस. सो व्हाट हैपेंस इज हम एक कैलोरी मीटर में जो स्पेस प्रोवाइड होती है उसमें हम अपनी टेस्ट ट्यूब को प्लेस करते हैं. हमारे पास होता क्या है, हमारे पास लाइट फिल्टर्स होते हैं. लाइट फिल्टर्स क्या करते हैं, लाइट फिल्टर्स ये करते हैं कि हमारे पास विद प्योर डिफरेंट लाइट्स होती हैं. हमारे क्या कहती हैं जो नॉर्मल वाइट लाइट होती है, हम कोई एक कलर चूज करती हैं और हम उस लाइट को यूज करती हैं. फॉर एग्जाम्पल यहाँ पर अगर आपको ब्लू फिल्टर या ग्रीन, फॉर एग्जाम्पल हम ग्रीन फिल्टर चूज कर लेते हैं. सो व्हाट हैपेंस इज दैट ग्रीन लाइट विल पास थ्रू आवर सब्सटेंस. हमने ये टेस्ट ट्यूब लेट से कलरीमीटर के अंदर रखी और कलरीमीटर में हमारे पास एक सोल्यूशन है. नाउ हमारे पास ग्रीन लाइट पास कर रही है. ऑब्वियसली अगर हमारे पास कोई भी कलर्ड सब्सटेंस है या कोई भी सोल्यूट किसी सोल्यूशन में प्रेजेंट है, सो हमारे पास जब भी लाइट उस टेस्ट ट्यूब को क्रॉस करेगी, some amount of light will be absorbed by the substances present within the solution. तो हमारे पास थोड़ी सी लाइट ट्रांसमिट करेगी. तो हमारे पास कलरीमीटर पर बेसिकली एब्जॉर्बेंस की या ट्रांसमिटेंस की रीडिंग आ जाती है कि हमारे पास कितनी एब्जॉर्बेंस ओवर द कोर्स ऑफ द रिएक्शन चेंज हुई है या कितनी ट्रांसमिटेंस ओवर द रिएक्शन चेंज हुई है. यूजुअली जो कलरीमीटर हमें वैल्यूज देता है वो एब्जॉर्बेंस की देता है कि हमारे पास एक सॉल्यूशन कितनी अमाउंट ऑफ लाइट एब्जॉर्ब कर रहा है. हमारे पास जितनी डार्कर सॉल्यूशन होगी उतनी ज्यादा एब्जॉर्बेंस होगी और जितनी क्लियर या लाइटर सॉल्यूशन होगी उतनी कम एब्जॉर्बेंस होगी. इसको सिंपली हम ऐसे अंडरस्टैंड करते हैं कि अगर मेरे पास एक ग्लास है, उस ग्लास से मैं वाटर फिल करूं और एक ग्लास में मैं कोका कोला फिल कर दूं, और अगर मैं क्या कहती है लाइट को एक्सपोज करूं उन दोनों ग्लासेस से, सो वील सी के क्लियर वाटर से सारी की सारी जो लाइट है वो पास कर रही है. जैसे कि जो हमारी कार विंडोज होती हैं, अगर कार विंडोज क्लियर है, ट्रांसपेरेंट है, तो बाहर की जितनी भी लाइट है वो अंदर आ रही होगी. लेकिन अगर हम टिंटेड विंडोज यूज करेंगे तो सारी लाइट अंदर नहीं आ रही होगी, बहुत कम लाइट अंदर आ रही होगी. सो इफ सोल्यूशन इज नॉट क्लियर, तो हमारे पास क्या कहते हैं उस सॉल्यूशन के कंटेंट्स लाइट एब्जॉर्ब ज्यादा करते हैं और कम लाइट उस सॉल्यूशन से ट्रांसमिट हो पाती है. डज दैट मेक सेंस? The student planned to follow the progress of the reaction for each concentration of lactase using a colorimeter. Explain why the student chose to use a colorimeter to follow the progress of the reaction for each concentration of lactase. So हमारे पास exact quantitative values आती हैं कलरीमीटर की रीडिंग पर, और हमारे पास क्या कहते हैं अगर हम कलर चेंज को अपनी आईज के थ्रू डिटरमिन करेंगे तो चांसेस हैं कि हमारे पास एरर ज्यादा हो. अगर हम एक कलरीमीटर यूज करेंगे सो दैट वुड एलिमिनेट एनी ह्यूमन आई एरर, तो हमारे पास ज्यादा एक्यूरेट रिजल्ट्स आएंगे व्हाइल यूजिंग अ कलरीमीटर. सो थ्रू अ कलरीमीटर quantitative results are obtained. Reducing human judgment errors if the student would have simply obtained conclusions by observing the change in color. The lactose in milk can be hydrolyzed using immobilized lactase or by using lactase फ्री इन सॉल्यूशन. तो हम इमोबिलाइज लैक्टेज बीट्स के थ्रू भी लैक्टोज हाइड्रोलिसिस कर सकते हैं या हम लैक्टेज फ्री इन सॉल्यूशन को भी यूज कर सकते हैं जिसको हम फ्री लैक्टेज बुलाते हैं. This results in milk and milk products that do not contain lactose and so are suitable for lactase intolerant people. तो अगर हम मिल्क में लैक्टेज बीड्स ऐड कर दें या हम मिल्क में क्या कहते हैं फ्री लैक्टेज ऐड कर दें तो हमारे पास जो भी मिल्क के अंदर लैक्टोज प्रेजेंट है the beads or the free lactase would hydrolyze it, resulting in a lactose-free milk or a lactose-free milk product to be formed, which then in turn are suitable for the use of lactose intolerant people. So जो भी हम lactose free milk का नाम सुनते हैं वो lactose free milk बनता ही हमारे पास lactase के hydrolysis के बाद है. Scientists carried out an investigation to compare the activity of lactase immobilized in very small magnetic beads, known as magnetic microspheres, with free lactase at different temperatures and at different pH values. Figure 3.2 shows the activity of immobilized lactase and the activity of free lactase at 5 different temperatures. Figure 3.3 shows the activity of immobilized lactase and the activity of free lactase at 8 different pH values. तो हमने पीएच पे दोनों की कैसी एक्टिविटी होती है और डिफरेंट टेम्परेचर्स पे कैसी एक्टिविटी होती है वो ऑब्जर्व किया. नाउ हम क्लियरली देख सकते हैं कि हमारे पास जो परसेंटेज एक्टिविटी ऑफ द लैक्टेज एंजाइम है इट इज बेटर देन द फ्री लैक्टेज. तो हमारे पास हायर यील्ड ऑफ हाइड्रोलिसिस प्रोड्यूस हो रही होगी. वन एडवांटेज ऑफ यूजिंग magnetic microspheres with immobilized lactase is that they can be easily recovered using an electric field and reused. तो हम immobilized lactase का जो एक फायदा है वो ये होता है कि हमारे पास हम उसको easily reuse कर सकते हैं because हम उसको recollect कर सकते हैं using a magnet. With reference to figure 3.2 and figure 3.3, explain why the results indicate that there are other advantages in using immobilized lactase instead of free lactase to produce lactose free products. So we can clearly see कि हमारे पास जो immobilized lactase है, the immobilized lactase has an overall greater yield compared to the free lactase at different pH as well as at different temperatures. We can also see that our immobilized enzyme is thermostable. Even at higher temperatures, its percentage activity is better than the free lactase. फिर हमारे पास क्या कहते हैं if we look at temperature, so at a range of different if we look at the pH graph, at a range of different pH, it is still giving us a higher activity compared to the free lactase. So हमारे पास जो free lactase है, the free lactase is giving us high activities in these pH, which is pH 6.5, pH 7, pH 7.5 and pH 8. But हमारे पास even pH 9 पर जो lactase immobilized lactase की activity है, it is comparatively better than the free lactase. So हमारे पास with respect to different pH values and with respect to different temperature values, the overall productivity or the overall yield of immobilized lactase is better. Let's move to the next question. Bacteria can be classified according to the type of cell wall they have. Gram-negative bacteria have a cell wall with an outer layer known as the outer membrane. Gram-positive bacteria do not have an outer membrane but have a much thicker peptidoglycan layer than gram-negative bacteria. Figure 4.1 is a diagram of a section through the cell wall of a gram-negative bacterium, which has an outer layer. With reference to figure 4.1, outline the similarities and differences between the outer membrane of a gram-negative bacterial cell and the cell surface membrane of a eukaryotic cell. So this is basically the outer membrane of your gram-negative bacteria. So similarities क्या है? Similarities ये है that we have phospholipids present. हमारे पास phospholipids present हैं. So like a eukaryotic cell membrane, phospholipids are present. Then we have membrane proteins present. So like a eukaryotic membrane हमारे पास membrane proteins present हैं. ये क्या हो गए? ये हमारी similarities हो गए. Differences क्या है? Differences ये है lipopolysaccharide is present in the outer membrane which we do not see in eukaryotic cell membrane. Furthermore हमारे पास क्या कहते हैं खाली मोनोलेयर प्रेजेंट है फॉस्फोलिपिड्स की, जबकि हमारे पास यूकेरियोटिक सेल मेम्ब्रेन में फॉस्फोलिपिड बाइलेयर प्रेजेंट होती है. सो हमारे पास मोनोलेयर का डिफरेंस है, और हमारे पास क्या कहते हैं, लिपो पॉलीसैकेराइड का डिफरेंस है व्हेन वी कंपेयर द आउटर मेम्ब्रेन ऑफ द ग्राम नेगेटिव बैक्टीरिया टू अ यूकेरियोटिक सेल मेम्ब्रेन. सो सिमिलैरिटीज, both have phospholipid molecules and transport protein. Differences would be eukaryotic cell membrane does not have lipopolysaccharides, and it has a phospholipid bilayer instead of a monolayer. Pathogenic and non-pathogenic gram-negative bacteria can produce extracellular vesicles. Figure 4.2 summarizes how two types of extracellular vesicles, OMVs and OIMVs, are formed. OIMVs are formed from the outer membrane and the cell surface membrane. So हमारे पास this is basically the cell surface membrane. This is basically the outer membrane. हमारे पास OIMV में क्या कहते हैं? We can see outer membrane भी present है और cell membrane भी present है. OIMVs can contain ATP and DNA. अगर हम OIMVs को compare करें OMV से, so OMV is only made up of the outer membrane. It is only made up of the outer membrane, whereas the OIMV is made up of the outer membrane as well as the cell membrane. हमारे पास क्या कहते हैं, plasmids, which are small circular DNA, are found in the OIMV, but they are not found in the OMVs. With reference to figure 4.2, suggest why fewer OIMVs are formed than OMVs. So हमारे पास ये comparatively कम form होती हैं. It is because we can see कि हमारे पास OMV form करने के लिए सिर्फ हमारी outer membrane क्या कहती हैं, fold हो रही है, और outer membrane eventually pinch off करके OMV बना रही है. On the other hand, OIMV बनाने के लिए हमारे पास सेल्यूलर कंटेंट भी चाहिए, हमारे पास सेल मेम्ब्रेन भी बल्ज करती है, हमारे पास क्या कहते हैं, जो पेप्टाइडोग्लाइकन सेल वॉल है, दिस इज द पेप्टाइडोग्लाइकन सेल वॉल, वो हमारे पास इस पॉइंट पर ब्रेक भी करेगी एंड इवेंचुअली फिर उसके बाद OIMV बनेगा. सो फॉर्मिंग एन OIMV इज अ कंपैरेटिवली मोर कॉम्प्लेक्स प्रोसेस because the cell membrane as well as the outer membrane has to bulge, and cell components also have to be a part of the OIMV. More complex process to form an OIMV, since the folding of both cell membrane and the outer membrane has to occur. Suggest why ATP is found within OIMVs but not in OMVs. हमारे पास ATP जो होता है, ATP एक बैक्टीरियल साइटोप्लाज्म में प्रेजेंट होगा. हमारे पास क्या कहती है, साइटोप्लाज्म में एटीपी प्रेजेंट होता है। द साइटोप्लाज्म्स एटीपी वुड बी ट्रांसफर्ड इनटू द ओआईएमवी, बिकॉज हमारे पास क्या कहती हैं, ओआईएमवी में सेल्यूलर कंटेंट भी प्रेजेंट होता है, साइटोप्लाज्म भी प्रेजेंट होता है। ऑन द अदर हैंड ओएमवीज में साइटोप्लाज्म प्रेजेंट नहीं होता। साइटोप्लाज्म अगर प्रेजेंट नहीं होता तो हमारे पास क्या कहते हैं, एटीपी प्रेजेंट नहीं होगा। ओआईएमवीज consist of bacterial cytoplasm in which ATP is found. Suggest and explain why the discovery that OIMVs contain DNA. कैसे इनके पास DNA है? Because they have plasmids. Plasmids are basically cytoplasmic DNA, circular cytoplasmic DNAs. Why the discovery that OIMVs contain DNA has implications for antibiotic resistance. हमारे पास antibiotic resistance क्यों होती है? हमारे पास antibiotic resistance तब होती है जब हमारे पास किसी भी बैक्टीरिया में म्यूटेटेड एंटीबायोटिक रेजिस्टेंस जीन आ जाए। हमारे पास अगर प्लाज्मिड में, जो कि साइटोप्लाज्मिक सर्कुलर डीएनए है, इफ दैट डेवलप्स an antibiotic resistant allele, it can transfer that allele to other bacteria, and thus it can be a reason of antibiotic resistance to spread. ठीक है? The plasmid within OIMVs can mutate to form a resistant allele which can then be passed to other bacteria by vertical transmission such as transformation. Clear? Any questions? Okay. Let's move to question number 6 and then wrap up this paper. Okay. So question number 6 states, in the pulmonary circulation of a mammal, deoxygenated blood becomes oxygenated when red blood cells pass through alveolar capillaries and hemoglobin within the cells combines with oxygen. The blood returns to the heart to be pumped around the systemic circulation. Describe the sequence of events occurring in the heart that allows blood returning in the pulmonary circulation to then enter the systemic circulation. You should include in your description the names of the relevant blood vessels of the pulmonary circulation and systemic circulation that are connected to the heart, reference to blood pressure changes that cause the opening and closing of valves. You do not need to include the details of the control of cardiac cycle. Okay, so basically, एक second, my tablet got disconnected. So when they're saying you do not need to include the details of control of the cardiac cycle, we basically don't have to talk about the sinoatrial node and everything. What we have to talk about over here is how the blood which is entering the heart is entering into the system. ठीक है, हमारे पास क्या कहते हैं, वो हमसे सिर्फ ये पूछ रहे थे, the sequence of events occurring in the heart that allows blood returning in the pulmonary circulation to then enter the systemic circulation. जिस तरह से हमारे पास जब हार्ट के अंदर ऑक्सीजनेटेड ब्लड आता है, वो कौन सी पल्मोनरी वेसल के थ्रू आता है? It comes via the pulmonary vein, and when it once it enters into the pulmonary vein, it then enters into the left atrium. When it enters into the left atrium, it then moves towards the left ventricle via the AV valve. हमारे पास जो AV valve है उसको हम क्या कहते हैं, bicuspid valve भी बुला सकते हैं, because we know कि left side में हमारे पास bicuspid valve भी होता है. Now हमारे पास जब bicuspid valve के थ्रू एट्रियम का ब्लड वेंट्रिकल में आ जाता है, then the ventricle's pressure starts to increase. When the ventricle's pressure starts to increase, the first thing which happens is that the AV valve closes to prevent the backflow of blood to the atrium. The ventricle's pressure increases. When it increases above the aortic pressure, the aortic valve or the semilunar valve in the aorta that opens. And once it opens, the blood from the left ventricle enters into the aorta, and then through the aorta, it is supplied to the rest of the body. Clear? Any questions? Figure 6.1 is a drawing of a hemoglobin molecule to show its globular structure. Describe the quaternary structure of a hemoglobin molecule. तो हमारे पास quaternary structure में क्या होता है जब भी two or more than two polypeptide chains associate via hydrogen bonding, hydrophobic interactions, ionic bonding और disulfide bonds, तो हमारे पास quaternary structure form होता है. हमारे पास hemoglobin की quaternary structure में we have four polypeptide chains, two alpha chains and two beta chains, which associate via these four bonds to form the quaternary structure. The hemoglobin molecule consists of two alpha globin chains and two beta globins, which associate via hydrogen bonds formed between the R groups of the four chains. Outline how a hemoglobin molecule can become fully saturated with oxygen to form oxyhemoglobin. So basically, a hemoglobin molecule has four heme groups. Um, हमारे पास क्या कहते हैं each heme group basically has an iron ion in the center. One oxygen molecule can bind to one iron ion. So a total of four oxygen molecules binding to the iron ion forms the oxyhemoglobin. Iron ion is present in each heme group of the four genes. One oxygen molecule can bind to one iron ion. A hemoglobin molecule can become fully saturated when all four iron ions are bound to four oxygen molecules. Okay. So that's it for this paper. We shall start with a new paper 2 tomorrow. Till then, take care. Allah Hafiz.